3 of those letters in a row specifies one amino acid. That's simply the coding for it. 3 of those letters in the DNA language codes for one amino acid. So a long sequence of DNA codes for the long sequence of amino acids you put together and thus make your protein. And what you've got at that point is a simple relationship. The string of DNA letters that codes for a single protein, that string of DNA letters is called a gene. A gene is the stretch of DNA that specifies one single protein. And you get a couple of complications there. There's a 3-letter DNA word which tells you we're at the beginning of a protein that starts a new one, and at the other end there's a different 3-letter one which is a stop sequence, tells you we're done with it. And that's the flow of information. This is the central dogma of life. This was a term that emerged in the 1960s after Watson and Crick came up with the structure of DNA and was worked on how the information flows. Information flows from DNA and its sequence of letters to ultimately protein and their function. And what you then have is a very sort of logical sequence. You've got your string of DNA, and you've got them thousands and thousands and millions and billions of these letters long. And each stretch of DNA that constitutes a gene then codes for a certain protein. And shown here in this diagram, we've got our looping or helical DNA. And you know if you magnify it enormously, there's going to be long strings of this 4-letter alphabet. And this stretch, beginning with its start signal and its stop signal, codes for
Continue
protein
on
A,
route.
which is shown here as a receptor. Next to it is protein B, coded for by gene B, in this case, a protein neurotransmitter. The next one is a channel. You remember back when calcium goes flowing into a cell to do that long-term potentiation stuff. There's got to be sort of this gate that opens up, which allows calcium to flow in. A calcium channel is the term. That's another one of those proteins. Stretch of DNA coding for 3 genes in a row, thus coding for 3 different proteins. And what you wind up getting is, just because there's such a staggeringly large number of genes, it winds up being a little bit cumbersome to have them all in one single long stretch. It simply evolved that you have your enormous DNA library broken up into separate volumes. Each separate one is called a chromosome. And it just picks up from there, and different species have different number of chromosomes, variability there. And what you wind up getting is, when you look at us, we've got about 30,000 different genes. And that's an enormous amount. Okay, so that's the flow of information from DNA to proteins. One extra small step in there, and this is one that simply happens, and this is how it's evolved.